Right now, as you read these words, light is entering your eye, striking a thin layer of cells at the back of your retina and triggering a chain of chemical reactions that your brain interprets as vision. This process feels simple, automatic, modern, but the machinery doing the work is not modern at all. The proteins in your retina, the ones converting light into electrical signals at this exact moment, are older than dinosaurs, older than trees, older than any animal alive on this planet today. Your eyes are using technology that evolved over 500 million years ago, and almost nothing about it has changed since. It started in the ocean. Before there were eyes, there were cells that could detect light, not see, detect. A faint chemical response to the presence or absence of photons. These cells used a protein called opsin, and opsin did something remarkable. When a photon of light hit it, the protein changed shape. That shape change triggered a signal. That signal told the organism something important. Light is here, or light is gone. This happened in creatures that had no brains, no heads, no faces. Simple multicellular organisms floating in ancient seas roughly 600 million years ago. During the Ediacaran period, they could not form images, they could not detect color or motion or edges. They could only tell the difference between light and dark, and that was enough. Because light meant the surface. Light meant food. Light meant exposure to predators, and darkness meant safety, or starvation depending on what you were. Even that crude binary signal, light or no light, gave these organisms an advantage, and evolution locked it in. The opsin protein that made it possible is still in your eyes right now. Not a descendant, not a cousin. The same gene family, performing the same function, using the same photochemical reaction it used more than 500 million years ago. Geneticist Todd Oakley at the University of California, Santa Barbara, has traced the evolutionary lineage of opsins across hundreds of species. What he found is staggering. The same ancestral opsin gene duplicated and diversified into the different photoreceptor types found across the entire animal kingdom. Your rods, which detect dim light, and your cones, which detect color, both descend from that single ancient protein. Every animal that sees, from eagles to octopuses to jumping spiders to you, uses opsins derived from the same molecular ancestor. Vision did not evolve multiple times from scratch. It evolved once, and then it branched. Your eyes and the eyes of a squid share a common origin that predates the split between vertebrates and invertebrates. That split happened roughly 530 million years ago, during the Cambrian explosion. The Cambrian explosion is when everything changed, in a span of roughly 20 to 25 million years. An evolutionary blink, animal body plans diversified at a rate never seen before or since. Armored creatures, jointed creatures, burrowing creatures, swimming creatures, all appearing in the fossil record almost simultaneously. And the leading theory for why it happened is eyes. Paleontologist Andrew Parker at the Natural History Museum in London proposed what he calls the light switch theory. His argument is that the evolution of the first true image-forming eyes triggered an arms race. Once one predator could see, prey had to evolve defenses, shells, spines, camouflage, speed, burrowing, and prey that could see the predator. Coming survived longer than prey that could not. So eyes spread, fast. And every body plan that followed was shaped by the pressure that vision created. Your eyes did not evolve in a peaceful world. They evolved in an arms race, and the stakes were extinction. The first true eyes in the fossil record belonged to trilobites. These ancient arthropods had compound eyes made of crystalline calcite, with hundreds of individual lenses packed into a curved surface. Their eyes were so well designed that some trilobite species could see in almost every direction simultaneously. Trilobites appeared roughly 521 million years ago. They are long extinct, but the optical principles their eyes used, refraction, focusing, light detection via opsins, are the same principles your eyes use today. Here is where it gets personal. You have 3 types of cone cells in your retina. One detects short wavelengths, what you perceive as blue. One detects medium wavelengths, green. One detects long wavelengths, red. These 3 cone types combine their signals to produce the full spectrum of color you experience. This system is called trichromatic vision, and most mammals do not have it. Dogs see in 2-color vision. Cats see in 2-color vision. Horses, cows, rats, rabbits, 2. Most mammals are dichromats because roughly 65 million years ago, when a mass extinction wiped out the dinosaurs, the small, furry mammals that survived were primarily nocturnal. They lived in the dark. Color vision was a luxury they could not afford. The genes for extra cone types mutated and degraded. 2 cone types were enough to survive the night, but primates went back to the trees. Your ancestors, roughly 30 to 40 million years ago, returned to a daytime life in forest canopies. And in the canopy, color matters. Ripe fruit is red or orange against a green background. Ripe fruit is calories. Calories are survival. A mutation duplicated one of the 2 remaining cone genes and shifted its sensitivity just enough to create a 3rd channel. Trichromatic vision returned. Not because your ancestors needed to see beauty, because they needed to find lunch. Neuroscientist Jay Neitz at the University of Washington has studied this mutation extensively. The duplication event happened on the X chromosome, which is why colorblindness is far more common in men. Men have 1 X chromosome, so a single defective copy means the 3rd cone type is lost. Women have 2 X chromosomes, so a backup copy usually compensates. Your color vision is not just a product of evolution. It is a product of a specific genetic accident on a specific chromosome tens of millions of years ago. But your eye's story goes deeper than color. Your retina is built backwards. Light enters the eye, passes through a layer of blood vessels, passes through a layer of nerve fibers, passes through several layers of processing cells, and only then hits the photoreceptors at the very back. The wiring is in front of the sensors. This is the opposite of what any engineer would design. It is like putting the cords in front of the television screen. Why? Because your retina evolved from brain tissue. During embryonic development, the retina forms as an outgrowth of the developing brain. It literally buds outward and folds into a cup shape. The photoreceptors end up at the back because of how the neural tube folds during this process. It is a constraint of developmental biology, not a failure of design, but it means every vertebrate on Earth, every fish, frog, bird, and mammal, has a retina wired backwards. This backwards wiring also creates your blind spot. The nerve fibers that carry signals from the retina to the brain have to exit the eye somewhere. That exit point where the optic nerve passes through the retina has no photoreceptors at all. There is a hole in your vision right now. You do not notice it because your brain fills it in, painting over the gap with a best guess of what should be there based on surrounding information. You are not seeing reality. You are seeing your brain's reconstruction of reality, assembled from 500 million-year-old photochemistry, filtered through backwards wiring, patched with a hallucinated fill for a hole that exists because of how a neural tube folded when you were an embryo. The mantis shrimp has 16 types of photoreceptors. 16. It can see ultraviolet, infrared, and polarized light. It perceives colors you do not have names for and never will. The pit viper detects infrared radiation through specialized pit organs, building a thermal image of the world, overlaid on its visible light vision. The jumping spider has 8 eyes arranged for 360-degree awareness. with 2 forward-facing eyes that provide telephoto-level zoom. Your 3 cone types and your single pair of forward-facing eyes are average at best. You cannot see ultraviolet. You cannot see infrared. You cannot see behind you. What you can do is see in extremely high resolution within a narrow central band, interpret faces and motion and depth with extraordinary precision, and process color in a way that lets you pick a ripe mango from a wall of green leaves at 50 meters. That is what your eyes were built for. Not to see everything, to see what mattered. Right now, 137 million rods and 6 million cones are firing in each of your eyes. Opsins are changing shape in response to photons, just as they did in eyeless organisms on the seafloor 600 million years ago. The signal travels from your retina through your optic nerve, crosses at the optic chiasm, and arrives at your visual cortex, where roughly 30% of your entire brain is devoted to processing what you see. You are using the oldest sensory technology on Earth. It has been patched, duplicated, rewired, and repurposed across 500 million years and millions of species. It is imperfect, it is backwards, it has a hole in it, and it is still the most extraordinary thing evolution has ever built. Right now, light is entering your eye. The same ancient chemistry fires, the same signal runs, and for a brief, flickering moment you see.